LuxMed CO2 Incubator, Tri-Gas Incubator, Hypoxia Chamber Incubator.
Welcome to LuxMed, a testament to the Make in India mission, a global force redefining health.
LuxMed proprietary software allows to download data from a CO2 incubator. You'll typically need to use software provided by the LuxMed. This software is usually designed to connect to the incubator via internal memory or CFR network via serial port, and allow you to retrieve, view, and potentially analyze the recorded data, send on email, save graph, PDF, Excel sheet. Also software get entry for all user information to get a printed report. LuxMed website, often in a download or support section, are supplied with equipment. Client screen for data logging. SMTP email setup and email formatting for report sharing configuration. User information and reporting data. Enter all test information. LuxMed CO2 Incubator maintains optimal conditions, particularly temperature, humidity, and CO2 levels, for cell and tissue cultures in a laboratory setting. It is a critical piece of equipment in various research and medical fields, including cell biology, in vitro fertilization, IVF, and pharmaceutical research. By precisely controlling these parameters, CO2 incubators create an environment that mimics the natural conditions of the human body, facilitating cell growth and development. LuxMed Tri-Gas Incubator is a specialized incubator used in laboratory settings to create a controlled environment for cell and tissue culture, particularly for experiments requiring precise control over oxygen, carbon dioxide, and nitrogen levels, in addition to temperature and humidity. It's often used in IVF, in vitro fertilization, labs, and for culturing various cell types, including anaerobic cells, and for hatching or germinating tissues. Principle. The core principle of a LuxMed Tri-Gas Incubator is to maintain a stable and precisely controlled atmosphere within its chamber, mimicking the natural cellular environment to promote optimal growth and viability. LuxMed Hypoxia Chamber Incubator creates a controlled, low-oxygen environment for cell and tissue culture, simulating conditions found in the body or in various disease states. These chambers are used to study how cells behave under reduced oxygen levels, which can be crucial in understanding various biological processes, including cancer development, stem cell differentiation, and responses to hypoxia-related diseases. Principle. LuxMed hypoxia chambers work by precisely controlling the oxygen concentration within a sealed chamber, typically by replacing oxygen with an inert gas like nitrogen. The core principle involves principle of operation. Temperature control. LuxMed CO2 incubators maintain a stable temperature, typically around 37 degrees Celsius, which is crucial for cell survival and growth. CO2 regulation. They regulate CO2 levels, often around 5%, which is essential for maintaining the pH of the culture medium. CO2 reacts with water in the medium to form bicarbonate, which acts as a buffer and keeps the pH neutral, around 7.2 to 7.4. Humidity control. LuxMed CO2 incubators also control humidity to prevent cell desiccation. Water pans or steam generators are commonly used to maintain a humid environment within the chamber. Air filtration. Some incubators include HEPA filters to remove particles and contaminants from the air, ensuring a sterile environment. Applications. Cell culture. LuxMed CO2 incubators are fundamental for growing and maintaining various cell lines, including stem cells, in research and biotechnology. In vitro fertilization, IVF, they play a vital role in IVF procedures, providing the optimal environment for embryo development. Pharmaceutical research. LuxMed CO2 incubators are used in drug discovery, vaccine development, and other pharmaceutical research areas. Tissue engineering. They are essential for creating artificial tissues and organs in tissue engineering applications. Diagnostic testing. LuxMed CO2 incubators are also used in diagnostic laboratories for various tests and assays.